Today, we delve into a series of fascinating and critical studies published in Animal Genetics and Genes, focusing on a group of devastating inherited neurological conditions in dogs, specifically the neuronal ceroid lipofuscinoses, in small Swiss hounds. Their research further underscores the heterogeneity of these lysosomal storage diseases, but also their shared core characteristic, progressive neurodegeneration. This particular investigation focused on two littermates, small Swiss hounds, who began exhibiting pronounced neurological signs around 12 months of age. The clinical picture was dire, characterized by progressive ataxia, which is a lack of voluntary coordination of muscle movements, as well as a significant loss of cognitive functions and vision. Tragically, the severity of these clinical signs necessitated euthanasia for both dogs just a few months after the onset of the disease, highlighting the rapid and devastating progression typical of many neuronal ceroid lipofuscinosis forms. Pathological examination of one of the affected dogs provided crucial corroborating evidence, revealing cerebral and cerebellar atrophy, indicating a shrinking of brain tissue in these vital areas responsible for thought, movement, and coordination. Furthermore, a definitive hallmark of neuronal ceroid lipofuscinosis, the cytoplasmic accumulation of autofluorescent material in degenerating neurons, was observed. This autofluorescent material represents the waste products that the cells are unable to process, leading to their buildup and subsequent neuronal dysfunction and death. These consistent clinical signs, combined with the characteristic histopathology, led the researchers to a tentative diagnosis of neuronal ceroid lipofuscinosis, setting the stage for their detailed genetic investigation. The pathological characteristics, including the intracellular accumulation of autofluorescent lipopigment and the loss of neurons, particularly in the cerebral and cerebellar cortices, are consistent with other forms of neuronal ceroid lipofuscinosis. Sometimes, atrophy of the retina is also observed. This study reinforces the commonalities across different forms of neuronal ceroid lipofuscinosis in dogs, demonstrating that a presumptive diagnosis can be based on evidence of heritability, progressive neurological dysfunction, and the accumulation of these autofluorescent storage materials in neurons.
Indeed, and the genetic investigation by Riedman's team provided the definitive diagnosis for the small Swiss hounds, classifying their condition as neuronal ceroid lipofuscinosis type 7, or NCL7. Using whole genome sequencing on one of the affected dogs, the researchers uncovered a significant genetic alteration, an intragenic duplication spanning 18,819 base pairs within the MFSD8 gene. This means a segment of the gene was unintentionally copied and inserted within the gene itself. Thank you for listening to the Veterinary Medicine Digest. If you're enjoying this, remember to like, share, and subscribe, as proceeds from this channel go directly to providing vital veterinary care for animals in need.